Welcome to the video series on biotechnology and molecular biology. I'm Pranav Doradla, and today we're going to be talking about proteins. So as I said before, today we're going to be talking about proteins. Some brief bullet points of what we're going to be talking about is on the screen. And in this first video, this first part, we're going to talk about that first bit that's circled over there. So proteins, in my view, are magnificent multi-purpose machines. They are essential to cellular function for a bunch of things, from membrane transport to cell-to-cell recognition, which is, cells have proteins on their outsides, and those proteins connect to each other and recognize each other. That's how your immune cells actually recognize um, antigens from invading viruses, like the coronavirus, for example. Reaction catalysis, so proteins are, there's some proteins called enzymes, which um, catalyze chemical reactions. They make chemical reactions go faster. DNA scaffolding, there's histone proteins and a bunch of different proteins involved in chromatin and compacting DNA, like we talked about in an earlier video. Structure, proteins help with membrane structure and with different cellular structure, and they're really important to for your bones, for your muscles, for everything in your body. Transcription, translation, obviously, you have enzymes that carry out those mechanisms, and there's a lot more that I haven't even mentioned here. So you can see on the slide, there's a picture of that, of all the different things proteins can do. So people generally look at proteins as having 4 layers of structure. The first layer is the primary structure, which is the sequence of amino acids. So if you haven't heard of this before, amino acids are the building blocks of proteins. And when they link up together, they form kind of like beads on a chain to make the protein. They're stabilized by covalent bonds between the amino acids. If you don't know what a covalent bond is, I would suggest that you spend some time online and look up some of the chemistry of this. You don't need to know a whole lot of detailed chemistry to understand this stuff, but the basics of bonding and inter and intramolecular bonding, like covalent bonding, is important. Then the next level is secondary structure. So that's at the local level, between an amino acid and its nearest neighbors in the peptide. There's different structures. So you can see on the screen there's alpha helices and there's beta pleated sheets. Those are the most common ones. Alpha helices are almost like the DNA helix. They're just a helix. They're a coil that's wound up. And beta pleated sheets are these strands which are kinked up and down, and there's multiple strands next to each other that are all hydrogen bonded together, so they form kind of a sheet. And there's other secondary structures as well, but these are the 2 main ones. Now, tertiary structure is when you have multiple secondary structures, multiple local secondary structures aligned in space, with regards to each other. So this is stabilized by hydrogen bonding, salt bridges, disulfide bridges, hydrophobic effect, van der Waals. So hydrogen bonding is something that I think you should look up on your own time to understand better. But basically, it's when hydrogen is covalently bonded to an electronegative element like fluorine, oxygen, or nitrogen. And then there's another place in another molecule or the same molecule that has that same layout. And then the hydrogen has a partial positive charge on it, and the electronegative element has a partial negative charge on it. So opposites attract, right? So the hydrogen is going to be attracted to the fluorine, oxygen, or nitrogen on another region of the same protein or a different protein. And the fluorine, oxygen, or nitrogen is going to be attracted to the other hydrogen. And this allows them to kind of be pulled together a little bit, but it's not as strong as an actual covalent chemical bond. Salt bridges are when you have fully charged parts of atoms that, or parts of molecules So fully charged ions that are attracted to each other. So you have like a, a -1 charge on an oxygen, and that's attracted to, let's say, a +1 charge on something else. And so this is stronger than hydrogen bonds because it's full charges, it's not partial charges, and this helps stabilize the protein as well. Disulfide bridges are actually a type of covalent bond. So this is when you have two cysteine amino acids. You can look that up on your own time, but they have, I think, a S connected to an H, and when two cysteines come together, the H's pop off. I think in a dehydration reaction, I'm not sure, and the S's then connect together in a covalent bond, and that's a very, very strong bond. So that allows different parts of the protein. So let's say you have one part of the protein here, one part here, different parts to come together and link up using disulfide bonds or disulfide bridges. Hydrophobic effect is the fact that in our cells and outside of our cells, even, it's full of water, right? Um, Water is just a very good solvent, and our bodies need it, so it's all over the place. And some amino acids like interacting with water because they're polar. If you don't know what polar is, again, I would suggest you review some basic chemistry online. You can find it on Wikipedia or Khan Academy. But some amino acids are polar, so they interact well with water because water is polar. Other amino acids are nonpolar, and that's why we call them hydrophobic. So hydro means water, phobic means fearing. So they fear water, the nonpolar amino acids. And because of that, they're not going to be on the outside of the protein where all the water is. They're going to go in the inside of the protein and get sequestered away from the water because they fear the water, right? And that's just energetically more favorable. So that leads to the protein kind of folding up, and you have a core of the protein where all the hydrophobic amino acids are. Van der Waals just refers to some of the London dispersion forces and other effects you have between hydrophobic amino acids. You can look that up on your own time. Now, quaternary structure, that's the 4, is when you have multiple peptides coming together to form one protein. So for primary, secondary, tertiary structure, these were actually peptides, not really proteins. The distinction is a little bit loose, but peptides are one single chain of amino acids. Proteins, on the other hand, are when you have multiple peptides that have all folded up, coming together to create an even larger protein. And you can see that on the picture there, where you have different peptide chains that are all folded up of different colors, so showing that they're not covalently bonded to each other, but they're just kind of glomming together. And that's why in many proteins you have these things called domains, which are different regions of the protein that are evolutionarily conserved across different proteins, and they have specific functions like binding a substrate for an enzyme, or causing the rest of the protein to move in some configuration, or something else. So there's lots of different proteins made, thousands or hundreds of thousands per cell, and they have all sorts of different functions, as we saw in the first slide, right? From structure to enzymes to a bunch of other stuff. And a cell might not need the same exact proteins 10 hours from now as it does now, because the cell is always adapting to different environmental cues and different cues from other cells and chemicals and all that stuff. So beyond simply changing rates of transcription and translation for proteins, we need ways to recycle and stop proteins from accumulating. So sometimes proteins can get damaged and accumulate, or they can unfold during stress or during certain diseases, even like Alzheimer's, for example. And an example of this is inclusion bodies, which you can see on the right, where many different proteins have kind of unfolded proteins have kind of just come together and formed like this mass. And we need ways to clean up the mess. So this is where protein regulation comes in. So there's the unfolded protein response and there's chaperone proteins. So when there's misfolded proteins in the endoplasmic reticulum, which is an organelle in cells that has proteins in it, and has the translation of proteins in it, there's 3 steps that take place. So the first step is once the cell has decided that, okay, there's too many misfolded proteins in the ER, it halts protein translation in the ER because you don't want more of those unfolded, nasty proteins to be created, and because you don't want it to form a mass, you don't want them to come together, get tangled up, and form an inclusion body, for example. Next, the proteins are degraded, so they're just like cut up. And then third, the cell calls for chaperone proteins to help. So chaperone proteins are also proteins, but they are exactly what they sound like. They chaperone the other proteins. They're big brother, and they help fold back together misfolded proteins during stress or disease, and even normal proteins. You know, in a normal situation, are folded by chaperone proteins because it just makes that process easier. And you can see that on the right over there, where you can see chaperone proteins kind of grab the protein and force it into the right conformation, into the right fold. So then they can go off and do their job. Now, sometimes proteins can't be folded the right way, and you just need to degrade them. So one of the most prominent ways to degrade proteins is called the ubiquitin proteasome pathway. So proteins are tagged with ubiquitin, which is a small chemical, and then a proteasome, which is a large complex of different enzymes and proteins, recognizes this ubiquitin tag, and then it slices up the protein. It literally cuts the covalent bonds between different amino acids, and turns the protein into a bunch of really small peptides, not even large normal peptides, but really, really small peptides of a few amino acids. And obviously then the protein has lost its function because it's not at all in the right structure, right? It's not connected anymore. It's just a bunch of free-floating amino acids and peptides, and it can't do what it was meant to do anymore. So that's all for this video. In the next video, we'll be going over part 2 of the proteins that you can see on the board there. And as always, happy learning.